Hello and welcome to another Mr. Carter Science Special. In this video we'll be looking at drug testing, how we would develop a particular compound which has potential medical uses and turn that into a drug that can be prescribed by doctors to treat disease. So first of all, let's consider what a good medicine is. Well, a good medicine must first and foremost be effective. It must prevent or cure a disease or treat a symptom. The medicine or the drug must be safe. It must not be too toxic or have unacceptable side effects. The drug must be stable. You must be able to use the medicine under normal conditions and store the medicine for a long time. The medicine must also be successfully taken into the body and removed from the body. It must reach the target organ and then be cleared from your body once it's done its work. You do not want these medicines to accumulate within your body because your body can't break them down. You might then reach toxic levels which could perhaps have a very negative impact upon your health. So the drug must be successfully taken into your body and successfully removed from your body. It can take up to 12 years to develop a new medicine, and it can cost up to upwards of 1,700 million or 1.7 billion, including failures and capital costs. So it's very, very expensive to develop new medicines. Any potential new drug must undergo a series of steps during the development process. And these steps are drug discovery, preclinical trials, clinical trials and licensing. Now with clinical trials, you'll notice there are 3 separate steps within the clinical trial process. So let's look at those steps in more detail. Firstly, drug discovery. Now I have discussed this in a previous video and I'll put the link to that video in the comments below. So drug discovery, a large number of chemicals are considered in the treatment of a specific disease. These chemicals will be tested on cells and tissues and organs in the laboratory. And we are testing to find for toxicity and efficacy. Will these drugs possibly damage and kill cells? And will the drugs be able to do the job we want it to do, its efficacy? If out of a large number of compounds, perhaps 10,000 compounds that go through the drug discovery stage, perhaps only 250 compounds will make it into stage 2, the preclinical trials. During these preclinical trials, there will be laboratory tests on animals to discover how the potential drug works in the whole organism. Possible doses are then calculated and potential side effects are discovered and evaluated. So out of those 250 compounds, perhaps 5 of them will be able to pass from stage 2 into stage 3, the clinical trials. So in a clinical trial, firstly, the healthy volunteers are given very low doses to check if the drug is safe and also to check for toxicity and side effects. Then, if the drug is found to be safe, the drug is tested on a small number of people with the disease in a double-blind trial to see if the potential drug treats the disease. So we're testing for its efficacy, and if the drug is found to be safe and effective, a larger double-blind trial will take place to find out the optimum dose. These trials are longer and with a larger number of patients, so the final set of clinical trials are far more reliable. The final step, where we might get just one compound approved, is known as licensing. If the drug passes all legal tests, it's licensed so your doctor can prescribe it, and the drug will be safely monitored for as long as it is used to see if there are any potential issues or issues that have arisen that scientists and doctors did not foresee. For example, if the drug has been used in a different way to the way that it has been prescribed. So what is a double-blind trial? Well, a double-blind trial is used to avoid the influence of the placebo effect. So what is a placebo effect? Well, the placebo effect is a psychological effect whereby a patient given a medicine by a doctor will often get better just because they believe that they're going to get better. So if we're going to have a double-blind trial and find out the effectiveness of a drug, we always need to compare and find out on how effective our new drug is compared to the effectiveness of the placebo effect. So all trials must be constructed as a double-blind trial. So in a double-blind trial, a group of patients with the target disease are randomly split into two groups. Each group will be very similar, with patients of a similar age ranges, similar gender mix, similar general health, similar heights, weights, BMIs, for a similar number of smokers, similar habits. And one of these two groups will receive the new drug, while the other group will receive a placebo. And that placebo is normally a sugar pill, a fake drug used as a control, something that we will compare back to. A control is always something you compare your results to see if you have a significant difference. So we're going to compare the effects of treatment versus no treatment, our placebo. The double-blind trial is known as double-blind because neither the doctors nor the patients know if they are receiving the real drug or the placebo drug. Only the person running the trial will know who is taking the real drug and who is taking the placebo. This way, the effectiveness of the real drug can be compared to the effectiveness of our fake placebo drug. In effect, we're comparing the effectiveness of our real drug to the placebo effect. If the drugs and the trials are successful, then the scientists will consider publishing their results. Scientific studies and their conclusions will be considered reliable because any investigation or study must be published in a peer-reviewed scientific journal. Peer review means that other scientists working in the field will have read the work and that they will agree with the methodology, the results and the conclusions, so that we know anything we read in a scientific journal is very reliable in its conclusions, and therefore we can have faith that the drug will work in the way that it's been described in the scientific article. So in stage 4, any potential drug after it's gained its licensing will be monitored for future use. And in particular, it will be monitored to see if the drug is used in a different way than it was originally envisaged and tested for. And the main example of when this has been a successful monitoring program is a drug known as thalidomide. Now, thalidomide was a drug used in the 1950s and 60s as a sleeping pill. However, it soon became known that the drug would prevent morning sickness, and pregnant women were often given this drug so that they would experience lesser morning sickness symptoms in early pregnancy. However, it soon became clear that pregnant women who were given the drug to prevent morning sickness gave birth to babies with limb deformities. So how did this happen? Why wasn't this tested for? Well, the drug manufacturers had tested thalidomide on animals in stage 2, but tests on pregnant animals had not been carried out and completed. Since then, drugs have had to be tested according to incredibly strict guidelines, which will include pregnant animals to see if the drug has any effect on the unborn offspring. So since then, as we've said, the drugs have to be tested according to incredibly strict guidelines, and thalidomide is now actually banned for use, but it is being considered and tested for a treatment of a number of diseases, including leprosy and cancers. However, this drug will never be allowed to be taken by women who are pregnant. I hope you've enjoyed this video on developing drugs. If you have, don't forget to like and subscribe and comment below to let me know what you've learned. Thank you very much for your time.